Alrighty, good morning, everybody. My name is Madison Ladines, and I am a senior here at Presbyterian College studying biochemistry. I did my research in the Department of Chemistry and Biochemistry under the advisement of Dr. Evelyn Swain. And today I'm going to be talking to you guys about the effects of complex sphingolipid pathway deletions on regulated cell death in Saccharomyces cerevisiae. So just to give you guys an idea of what we're going to be discussing today, we are going to start with the abstract and go into the introduction and talk about why we use yeast, what is the cell danger response and regulated cell death, what are sphingolipids, and then we're going to bring that all together and go into the hypothesis before we head into materials used for the various experiments we conducted, and then results and discussion of those experiments and conclusions that we drew from them. And then there will also be time for questions at the end. So to start off, we have our abstract. So regulated cell death, RCD, is in yeast and is homologous to cell danger response in humans, CDR. And sphingolipids are important signaling molecules and structural molecules, and we want to see how alterations in the pathways used to create complex sphingolipids affect regulated cell death in our cells under regulated cell death induced by acetic acid. So why do we use yeast? Seems like kind of a funny thing to be using. So yeast are actually excellent models of human metabolism. They're relatively inexpensive. They replicate very quickly. They have a fully sequenced genome, and there are also many mutants available, so that anything you could want to do or look at or observe in these model organisms may be readily available already. And if it's not, it is pretty ethically unproblematic to use yeast for experimentation. So now to dive straight into cell danger response, or CDR. Here is a diagram of what CDR looks like in the cell. So cells typically exist in what we call wakeful activity and nutrient uptake. So if all is well, the cell is functioning normally and at its best efficiency. It is, in terms of humans, eating and drinking as it should. Things are working properly. However, when it is faced with a stressor, internally or externally, it goes into what we call CDR, cell danger response. And so it moves out of that wakeful activity into CDR. And CDR is a cyclical multi-step process, and it goes from CDR 1, 2, and 3. If you go back here to the abstract page, you can see in this diagram the cyclical fashion of cell danger response and how it just keeps kind of turning. And as you go from wakeful activity to CDR 1, 2, and 3 and go back, you just kind of devolve down this little turn. And so the main goal of cell danger response is to recoup that cell, get it back to wakeful activity, and let it go on its way. However, sometimes our cells become stuck in these different stages, CDR 1, 2, and 3, or while in the cell danger response, they die. And so there are a couple outcomes to come from cell danger response. It is identifiable by multiple characteristics, and it's also largely controlled by the mitochondria, which is going to be important later in our discussion. So this is a more complex version of what I was just speaking to you guys about. So cell danger response, multi-step, cyclical, you can see that here. So you start in wakeful activity. The cell can use oxidative phosphorylation, aerobic glycolysis, and glycolysis as metabolism methods, and it can do that as it's needed. It goes integrated back and forth between what is necessary to keep that cell at its best fitness. However, when a stress, new information, injury, toxins, anything like that, a pollutant, is introduced, the CDR switch is flipped to on. And so with that CDR flip on, you have a few choices right off the bat. You could have your cell die, or it could go into restorative sleep, which kind of lets it skip ahead in the line and go straight to the end of the CDR response and head almost directly back into the health cycle, where it's in wakeful activity. However, if it doesn't do that and it heads into CDR1, you're going to go into what's called the containment and innate immunity stage. And glycolysis is actually the typical metabolism method for cells in this stage. And so it'll keep going through CDR until it hits checkpoint 1 unless there's excessive metabolic damage. And if there's excessive metabolic damage, that is also another place where you will see cell death through apoptosis or necrosis. However, checkpoint 1 divides CDR1 and CDR2. You hit checkpoint 1 and you can either head into CDR2, which is the proliferation stage where aerobic glycolysis takes precedent for your metabolism, or if there is significant mechanical strain or excessive DNA damage, you can have scarring or granuloma, or your cells can go into senescence. And so what both of these things do is that they make it so that your cells do not operate with its utmost efficiency. Scarring could interrupt communication in a tissue, and senescence is when your cells are not dead, they are still alive, but they are not actively dividing. And so they're kind of just sitting there in the way, which presents, communication issues within your tissues. So if you continue through CDR2, you will hit checkpoint 2. Checkpoint 2 is what divides CDR3 and CDR2. When you go into CDR3, if your body and your cell does not realize that it is coming out of danger and that it is healing, then it will have a persistent release of molecules that are used for defense. And so that can be where we see the rising of autoimmune disorders, chronic pain, neurological and psychiatric symptoms. And so that's not particularly seen as positive. Also, I forgot to mention, and CDR2 proliferation is the name of that stage. And that is where we see things like cancer, where cells grow without control or checkpoints. And so we would like to get cells out of that and let it keep going. And as I said before, when cells get stuck in these stages, that is where we see things with chronic pain, cancer, because the cells can't seem to get out of those stages. Back to CDR3 though, you are in the differentiation and development stage. And so you're not quite out of the woods yet, but your cells are recouping. They're almost there. They're using oxidative phosphorylation for metabolism, and they are headed towards checkpoint 3, which divides wakeful activity and differentiation and development. And if they pass through this checkpoint, they're back into wakeful activity and are considered completely healed. And again, that is what we want. We want our tissues and our cells to completely heal. However, we see problems when they get stuck along the way. And so, regulated cell death. I just told you guys about cell danger response. That's found in humans. I said we were going to talk about yeast. So how does this all connect? Well, regulated cell death is the connector here. Regulated cell death is the parallel pathway to CDR found in yeast. It shares similarities with apoptosis, and to be completely honest, it is not the perfect parallel pathway for CDR, because the end result of regulated cell death is cell death. Whereas with CDR, you can usually recoup those cells so that they are living and active again. Regulated cell death ends in cell death. And so the process in general is to produce healthier colonies by killing off the cells that are inhibiting the overall fitness of those colonies or of the environment. Regulated cell death is induced by outside stressors. It also can be induced by internal stressors, and much like CDR, is under a lot of mitochondrial control. At the bottom here, you see our mitochondrial morphology scale. You can see that mitochondria types are assigned to each stage of CDR based on their morphology, and it is on a spectrum, and this will become really important later when we're tying what we observe in our experiments to CDR and regulated cell death. So sphingolipids, I mentioned complex sphingolipid pathway in the title. What is a sphingolipid? So sphingolipids are structural and signaling molecules. They are used throughout the cell, anything from the cell membrane to really complex signaling cascades. And regulated cell death is no exception. Sphingolipids are constructed of a sphingosine backbone, a fatty acid bound to amide bond, and a head group. And so if you look at this diagram here, here's our sphingosine backbone. Our fatty acid is bound with this amide bond, and then the X in blue here denotes the head group. And so the X in the fatty acid, or the head group in the fatty acid, are really important for diversity. So our diversity arises from being able to change out that head group for different things, and from getting to elongate, shorten, or oxidate our fatty acid tails so that they look different, and can serve different purposes within the cell based on its needs and which pathway it's headed to. Regulated cell death is tied to sphingolipids. It is not characterized well, though. It is most characterized regarding the ceramide family, in which the head group is a singular hydrogen. So all of that goes into our hypothesis. So that's all tied together, and we want to look at how deletions of specific enzymes within the complex sphingolipid pathways that produce these complex sphingolipids for the cell, how they affect viability and phenotype in cells undergoing regulated cell death induced by acetic acid. And so what we are looking at specifically is, let me move myself so you guys can see that title. Complex sphingolipid pathway enzymes of interest are SIR4, CSG2, and ISC1. Elongation of fatty acids used to synthesize complex sphingolipids is what SIR4 does. So it's a part of that elongation process that creates those long fatty acid tails that are used to produce our complex sphingolipids. CSG2 is used further down in the process, and it synthesizes MIPC from IPC using GDP mannose. And then ISC, one degrades, it's on the flip side of all of this, and it degrades our complex sphingolipids to their components, which are ceramides, polar head groups, and long chain bases. Here is a diagram of our complex sphingolipid pathway. And this just gives everybody a visual of where these things are located, and what is up and downstream of them and how they are related to each other. So here's SIR4, located in this elongation cycle, produces this fatty acid to create phytoceramide. And then phytoceramide is used to create our complex sphingolipids, this whole group right here. So if you were to delete SIR4, as one of our mutants has, you would have a lack or an inefficiency in that elongation cycle, which would result in reduced phytoceramide and eventually reduced complex sphingolipids, because you don't have the correct amount of phytoceramide to produce all of your normal complex sphingolipids. CSG2 is located here. You have inositol phosphoceramide, IPC, and using CSG2, two other enzymes, and GDP mannose, which is a catalytically active carbohydrate, you are able to produce MIPC, which is mannose inositol phosphoceramide. Those are both complex sphingolipids, and so if you were to delete CSG2, you would not only have a decrease in the amount of MIPC and mannose inositol phosphoceramide too, but you would also have a buildup of phytoceramide, which is a little bit up here because all of your IPC wouldn't be being converted. ISC1, like I said, is a degradative enzyme. And so once you have your IPC, MIPC, and MIP2C, you can degrade it using ISC1 into their components, which are polar head group ceramides and long chain bases. And so if you were to delete ISC1, you would also have a buildup of complex sphingolipids. And you would, in turn, have a reduction of ceramides from ceramides are produced here, but also here. And so if you were to get rid of the degradation, you would have a decrease in the total amount of ceramides available to your cell. So before we go into our experiments, we're going to look at the materials that were used for these experiments. Acetic acid was used to induce regulated cell death, and selective galactose media was used to, set at pH 3, was used to produce a high number of mitochondria. And so how that works is that galactose is not the best option for yeast to metabolize. And so they have to work a little harder and produce a higher number of mitochondria, which makes it easier for us to observe and qualify. D399 and DIOC6 dyes were used. These are both fluorescent dyes. D399 looks at ROS, reactive oxygen species, specifically peroxides and hydroxyls. And then DIOC6 is used to stain mitochondria. And earlier I said that the mitochondrial morphology scale would be important, and you can see where that's going to tie in just a few minutes. And then YAPD plates, because here in the biochemistry department, we use YAPD plates for everything. So our first experiment is the ROS staining using D399. You can see the top picture are white light photos and the bottom picture are fluorescent photos. The vacuoles can be seen as well as the cytosol, and that can then be reflected here. The ROS staining, again, is picking up reactive oxygen species within these cells. So where there's a higher or brighter color is where there's more ROS. This specific set of data was not labeled properly after these photos were captured, and so I cannot tell you which these are. However, if you come over here, the SIR4 induced with acetic acid can be seen. You see these odd vacuoles. Something that is common in SIR4 mutants is that we see that they have fragmented vacuoles, and so you can see these odd vacuole formations. But then if you come down here to fluorescence, you can see that our ROS is not within our vacuoles, and there's also not dye turnover into our vacuoles, and so all of this ROS is located in the cytosol. This was one of our better pictures, and we did have some trouble getting pictures that were stained strongly enough for us to qualify and quantitate. So our next experiment is our mitochondrial staining experiment using DIOC6 fluorescent dye. All of these images are experimental. So this means that we use acetic acid to induce regulated cell death in all of these cells. You can see here, there is the staining of the mitochondria. It's all outside of the vacuole. And if there is any in the vacuole, we believe that would be turnover. And so with acetic acid, the SIR4, we can see these small punctate mitochondria. They're very individual. The resolution is not excellent. And then for CSG2, we were able to see all of the mitochondrial morphology. So you can see how these are kind of connected, but there are also punctate mitochondria around, depending on which cell you're looking at. Wild type, we saw some small punctate mitochondria here, all throughout here. And then ISC1, we also saw some filamentous, which was interesting. You can see how they're kind of connected. It looks a little bit like a web. You can see that here as well. So our next set of data is from our control. So these were not induced with acetic acid, and we wanted to see if there were differences in the mitochondrial morphologies based on whether or not they'd been induced with acetic acid. You can see here in our SIR4, we have filamentous. They are all kind of connected and look almost woven. Here you can also see all of them again. This was really interesting to us. We have some punctate, there's some filamentous up here, some punctate. And then in these other two, we can also see some filamentous. You can see how it's like a line, it's connected. These are connected together. And our last experiment was plate viability with acetic acid. With acetic acid or without, what we did was we grew up all of our strains, including wild type, in YAPD media overnight. We then grew them for one growth cycle in selective galactose media set at pH 3 with or without acetic acid. And then we diluted them and plated approximately 400 cells to each plate so that they could be quantified. And so that's what you're seeing here is those plates that had grown for about 72 hours. Each strain was done with and without acetic acid. And so these are the results that we were able to glean from those plates. We counted all of our CFUs, or colony forming units, which if you go back here, this is a colony forming unit. It's just a single colony that is thought to originate from one clone. And so we used our wild type as our control, wild type without acetic acid induction. And then we're able to calculate the viability percentages of each of our strains based on their condition as well, so with acetic acid or without. So what does all this mean? Why is this important? We drew some conclusions from our experiment. So for our plate viability, we're able to see that SIR4 had overall poor viability under regulated cell death conditions. And so if this is our control, SIR4 is greatly reduced. And you can see here, if we were supposed to have 717 colony forming units, SIR4 under regulated cell death conditions only had 149 colony forming units. What is interesting, though, is that we were also able to see that CSG2 had increased viability under both conditions as compared to our control. You can see that here, if there were supposed to be 717, we had above that number in both acetic acid plus and minus for CSG2. And then all of our deletions, except for CSG2, obviously, have reduced fitness under both regulated cell death conditions and control conditions. You can see that if this is 100%, these are significantly lower, SIR4 being by far the lowest. And then even wild type induced with acetic acid had a slight reduction in viability. The raw staining, as I said before, we did not have properly labeled samples the first time, and then the second time our staining did not take well, and so it was difficult to qualify those results, and so those results were inconclusive. Our conclusions for our mitochondria staining, though, were interesting. As I pointed out on the slides with the fluorescence, you could see the different morphologies, and so without acetic acid, we saw small punctate in wild type in SIR4, filamentous with ISC1, and all mitochondrial morphologies for CSG2. For acetic acid induction, we saw filamentous in wild type SIR4 and ISC1, and then again, CSG2 had all of our mitochondrial morphologies, which was really interesting. And so this diagram of mitochondrial morphology was expanded so that we could then look at these results and kind of qualify where these cells might be falling along the CDR cycle according to their mitochondrial morphologies. And so you can see the small punctate, large punctate, and filamentous here at the bottom. And so the SIR4, ISC1, and wild type were all filamentous with acetic acid. And so that could point to them being in CDR3, whereas CSG2 had all mitochondrial morphologies in both conditions. So we had cells in all of these, which is interesting because it also had an increased viability. And so there could be ties there. And then for the non-acetic acid induction, we could see that maybe these were in CDR1. And again, this is at the very beginning, and so we're not sure how far the mitochondria extend into wakeful activity for punctate and freestanding. And then filamentous for ISC1, so maybe it could be in CDR3. Another thing that we saw were circles. These were just circular morphologies that we saw within ISC1 and CSG2. We're not really sure what they were, but we did notice them in both of these strains. And our hypothesis is that it could be from budding scars, it could be scarring from stress, and that would be something that we could look into further. And speaking of further research, what could we do with this research? We would need to redo our Ross experiment and hopefully produce results so that we could see the different Ross levels under different conditions of regulated cell death or not regulated cell death induced by acetic acid. We would like to replicate our experiments that are already done to confirm our conclusions or further our thinking on all of them. It would also be interesting to look at mitochondrial permeability in relation to all of our deletions that we're working with now, mitochondrial permeability has been seen to play a role in apoptosis. And so should these deletions have effects on mitochondrial permeability and ultimately apoptosis or regulated cell death, we would like to know. And then something else we could do is examine effects of deletions within the same pathway. So if you flip all the way back to the pathway here, what would happen if maybe we mutated, if we deleted SIR2, or if we deleted an LIP1 or IPT1? How would that affect sphingolipid production and ultimately regulated cell death and CDR? So these are the references that I used for this presentation. A special thank you to the Lord for giving me the ability to be able to present this and be a part of this community that I've found here at Presbyterian College. Special thanks to my family and friends and everybody, especially that's here, Dr. Swain and the chemistry and biochemistry department at PC. And now we have some time for questions. If you have any questions, put them in the comments below. And I will respond and hopefully give you some answers. Have a great day, guys.